Okay, so welcome everyone to the Climate Festival. My name is Natasha Barbolini. I'm a paleoecologist here at Stockholm University, and I'm going to talk to you about the biggest mass extinction that the Earth has seen in the last 541 million years. So first of all, I'm going to give you a little how-to guide on how to kill all life on Earth, or almost. So first of all, we add lots of greenhouse gases. These increase temperatures a lot. Then we add some heavy metals and pollutants. Let's throw in some ocean acidification. Don't forget sea level changes, burning of vast reserves of coal, the ozone layer breaking down, and widespread deforestation and extinctions. So can anybody tell me what I'm talking about? Any ideas? The things we're doing right now, okay? I love that answer, but I'm not going to talk about that today, although I'm going to show you how similar these two events could really be. So first we'll talk about what is a mass extinction, okay? Well, species evolve and change and go extinct all the time, okay? This is not something unusual. It's called the background rate or the background level of extinctions. And so this is normally in response to environmental changes that are happening. Natural selective pressures force these animals to evolve, or if they can't adapt, they go extinct. But mass extinctions happen when these changes are too big or too fast for most species to adapt to in time. And so instead, they go extinct and they don't contribute any further to the tree of life on Earth. So, for example, if your grandmother didn't survive long enough to give birth to your mother, you wouldn't be around today because your ancestors have gone extinct. And this is what these creatures did. They didn't contribute any further to life on Earth. And what we know is that extinction is almost certainly forever, okay? It's very final. So this is great. It made a great book and a movie, but Jurassic Park is not going to happen anytime soon, even with our advances in genetics. So we can wipe that off the table. So how do we know what a mass extinction is? Well, in the past, we've identified 5 big mass extinctions. And if you look on the left-hand side of the graph, you can see the number of families. This is one way to look at biodiversity levels in the past. And at the bottom, you can see the amount of time in millions of years that the Earth has gone through. So this is just the last 600 million years. Of course, Earth is much older than that. But this shows the number of families for this time period. And what we find is that there were 5 major mass extinctions over the last 600 million years. So I'm not going to talk about the much more famous one, which is the one that killed the dinosaurs through a bolide impact. Okay, most people know about that extinction. I'm going to talk about a different one. Okay, it happened much earlier in time, and it's called the Permo-Triassic extinction, otherwise known as the Great Dying, because so many species were lost. And so this happened 251.9 million years ago, so a really long time ago. And we lost an incredible amount of diversity on Earth. So if we didn't lose the dinosaurs, in fact, they hadn't even evolved yet, what did we lose? Well, this is a reconstruction of what a Permian world may have looked like. So the creature on the left here is called a pariasaur, and this may have been an ancestor of modern turtles. And the thing that's attacking it is a gorgonopsian. So this is not a dinosaur, okay? It's actually more related to mammals. And this group may have given rise to modern mammals today. And they are known as the mammal-like reptiles or synapsids. So the plants, okay? We also lost a lot of plants. Although many of the same groups can be grown in a greenhouse today. You can see things like ferns, conifers, ginkgos. These are all familiar, right? But we also lost some other plants that didn't survive at all, and we have no similar groups to them today. And one such plant is called Glossopteris, and this was a plant that existed through almost the entire Permian. So you can see Glossopteris there in the top right-hand corner. This is what this plant may have looked like, and the fossil leaves that we find look like this. So the name Glossopteris comes from the leaves being tongue-shaped, and they're really characteristic. You can recognize them in fossil deposits of the Permian across the entire southern hemisphere. So this is a reconstruction of what a plant may have looked like. You had large trees, but you also had some smaller shrub-like growth, most likely. And these plants stretched over almost the entire southern hemisphere during the Permian period. So during this time, the continents were joined into one big landmass that we call Pangaea. And you can see that Glossopteris, in the dark green arrows there, covered most of South America, Africa, India, Australia, and Antarctica. And this plant group went entirely extinct at the Permo-Triassic boundary. So how do we know about these past extinctions that happened so long ago? Well, I'm a paleoecologist, and paleontologists are people who study past life and ecosystems on Earth. And we look at the geological record to find these fossils, and they tell us about biodiversity levels at different times in the past. So we get all sorts of fossils, plants, animals, and invertebrates. And you can see on the left-hand side of this time scale here, okay, many different kinds of creatures have existed at different times in Earth. So we know that mass extinctions have happened when we find particular groups or lineages that don't continue past a certain boundary. So we find them in rocks of a certain age, but we don't find them above those rocks. And mass extinctions are places where many different groups all went extinct at the same time. So we know that something bad was going on there, right? So the Permo-Triassic extinction in particular, what happened, okay? Well, it was the biggest in the last 541 million years. It was much, much bigger than the famous extinction that killed the dinosaurs. So when I say most dinosaurs, we actually do have some dinosaurs still living with us today. Fun fact. Can anyone tell me what they are? Chickens, exactly, okay? Birds. Birds are the last living groups of dinosaurs left. And paleontologists already believed this because of the similarities in the skeletons that you see between modern birds and dinosaurs. But when they found these dinosaurs with feathers on them in China, then the case was pretty much sewn up, is birds are in fact dinosaurs. So fun fact for today, but let's go back to the Permo-Triassic extinction. So this was really serious. It killed about 81% of marine species, about 75% of the vertebrates on land. And we lost all of these groups forever. So what caused this? Well, basically volcanoes. So we had a huge volcanic system in northern Eurasia called the Siberian Traps. And when these volcanoes exploded, they released huge amounts of greenhouse gases and nasty things like halocarbons into the atmosphere. So an example of a halocarbon is CFCs, which destroyed the ozone layer last century. And so all these horrible gases at these high northern latitudes went into the atmosphere, and they may have set off a huge chain reaction with a bunch of different effects. And you can see this diagram is really complicated. But things like breaking down the ozone layer, acid rain, volcanic darkness, carbon dioxide emissions, global warming, all these things probably work together in order to cause such a huge extinction. And what we really want to know more about now is this particular ozone depletion, which could have caused really increased UVB levels or ultraviolet levels. And there's a lot of knowledge gaps surrounding how the ozone could have broken down during this extinction. But how do we know about the ozone layer? I mean, that's air, right? So air doesn't make a fossil. Like, how would we know about that? Well, some of my colleagues who study fossil pollen, like I do, they found really weird looking pollen in these rocks, which preserve the Permo-Triassic extinction. And so instead of looking like normal pollen there on the top right, they were really strange. They had like 3 Sacchi, or they were completely mutated. And when my colleagues looked at pollen from the Chernobyl radiation disaster that happened in 1986, they saw that the pollen looked pretty much identical to this pollen from the PT extinction. So then what they did was they experimentally blasted some living pine trees with really powerful UV radiation. And what did they find? Exactly the same kind of malformed pollen. And so this probably records a huge increase in radiation 251 million years ago. And this would have caused widespread forest devastation and also skin cancers and all sorts of other terrible mutations on Earth. So just as important to know about how all these organisms died is how did life recover on Earth? How did we come back from such a big extinction? Well, we know it took a really long time, and that's because conditions were really hostile to life for a long time afterwards. We see that coral reefs and forests took about 20 to 25 million years before they started to form again. So that's a really, really long time. And we aren't sure yet how long the ozone layer took to recover. So this is a hot new topic of research. What we do know for sure is that animal life, both on land and in the oceans, was completely different before and after the extinction. So this extinction changed the trajectory of evolution on Earth completely. So why should the past matter today? This happened 251 million years ago, right? So why is it relevant? Why does it matter? We should just leave it in the past. Okay, well, on the right-hand side, you can see our temperature increases for this century compared to the previous period. And the red, of course, is the most, going down to the blue. So for most of our world, we've really increased the temperatures, as you know, in a very short period of time. And what we know is that the climate models to predict our near future, used by the IPCC, for example, they tend to underestimate climate change compared to the geological record. So if you actually look at what happened in the past compared to what these models forecast, potentially the effects could be more serious. And we know this is partly because Earth responds quite slowly to some changes, like glaciers melting and permafrost thawing, deep ocean warming, or vegetation restructuring in ecosystems. These changes all take a really long time. We won't see them in the next century, but they will continue to have an effect on our climate for many years to come. So because of these changes, some people have said that we could already be entering a 6th mass extinction. So we've had 5 big ones in the past. Are we already in the middle of a 6th one? Well, let's look at the evidence. So, to know if this extinction is really happening, we have to know if it exceeds the background rate. So this is the normal rate at which species tend to go extinct over the entire period of geological history. And there's 2 main factors that control this. First, the speed at which organisms are going extinct, and secondly, the magnitude or the severity, or how bad did it really get? How many species are we losing? So if we look at levels today, we find that the rate of species extinctions, how fast it's happening, is much more similar to intervals of mass extinctions in the past. It's not similar to intervals in between them, where not as many species went extinct. In terms of magnitude, we know we are losing about 100 to 1,000 times more vertebrate species than in the past right now. So both in terms of rate and magnitude, it seems that we really do qualify for entering the 6th mass extinction at this time. And these factors do not take into account something else, dead clades walking. So what you'll see after mass extinctions is there'll be some lineages that will survive the extinction, but then they'll go extinct really soon afterwards. And that could be partly due to relationships that they have with other creatures in the ecosystem. So for example, bees are going extinct today. So what will suffer once bees have gone extinct? The plants, right, that pollinate them. And then us, right? We eat the food that the plants make. So all these things could be dead clades walking, including maybe us. Scary thought, right? So what can we do about it? Well, we need more science to refine the details, but we already know more than enough to act. And so it's really important, okay, to take action. The Perma-Triassic already holds a lot of important lessons and warnings about when one enters a mass extinction, what we can expect. And what we know is that if it took 20 million years for ecosystems to recover after the PT, that's about 200,000 generations of humankind before ecosystems could be back to normal. What we also know is that the climate, the warming, and the ecological crisis are intertwined, and we have to deal with them together. If we only act on warming, we're not going to solve the problem. And if I bring you back to the first slide that I showed you with all these different points, if we only deal with the first two, these greenhouse gases and increasing temperatures, we're going to miss all the other things below that caused the Perma-Triassic extinction and that are really similar to what we're doing today. So I'm going to ask you guys, do you think we're recreating a Perma-Triassic extinction today? What do you think? Yes? No? Undecided? Okay, well, now you can ask me any more specific questions about the PT extinction that you may have. Thank you. Questions? Yes?
You said the number of species going extinct were 100,000 times more than the last extinction, or like the more normal rate?
You are asking whether 100 to 1,000 times vertebrate species losses is more than what we had in the past or more than the previous extinctions. These are the biodiversity levels that we inherited. So before humans started to change the climate. So we've used the normal background rate of extinctions that we could calculate from animals that existed on Earth pre-Holocene or pre-humans, okay, when humans started to really modify the climate at about 10,000 years, not the levels of a mass extinction. Okay, so these pretty much compare with levels for other mass extinctions that we have seen in the past.
Yeah, so you said that the Siberian melting caused a large release of halocarbons in the atmosphere, right? Where did those halocarbons end up after the extinction? Are they like still trapped in the ground, or are they gone to the ocean?
Right, so you're asking about the halocarbons, and once the Siberian traps released them, then what happened to them in the environment? So these halocarbons probably took a long time to become sequestered away, okay? For example, things like methane exist as hydrates that are frozen in the deep ocean floors, and warming can release them. But this is one way that they can be sequestered back. So over time, ecosystems could sequester away these really nasty chemicals so that they didn't affect life the way that they did over the mass extinction. But it took a really long time for the ecosystem to do that with certain chemicals.
Right, that means that there are like large deposits of chlorine and derivatives right now. Right?
Yeah, there are. So, but they're just not being released. For example, mercury, there are really large stores in the permafrost, and these are being released as the permafrost thaws. Yeah. Questions? Yes. Can we see individual differences now? Sorry, can we see? Right, so you do see different levels of biodiversity losses in different areas, and the areas of losses are the greatest in the tropics right now. But that doesn't mean that what's going on in the tropics is the worst. It's just that biodiversity is really high in the tropics to begin with, and primarily that there's a lot of deforestation that's happening in tropical areas, like in the Amazon, for example. And then you lose a lot of species with deforestation and with land modification. So yes, there are areas where we are losing more species. Time. Great, thanks very much.
Good morning. Let's come back. I'm going to talk about something a little bit different. And my name is Sonia Merinhero, and I'm a researcher at the Department of Ecology, Environment, and Plant Sciences at Stockholm University. And today I'm going to talk to you about how will the green jewels of the forest will cope with climate change. And you might be wondering, who are these green jewels? And of course, you're right if you are thinking about mosses. Mosses are a very large group of very tiny plants that they live almost everywhere on land. There are approximately 12,000 species in the world, and as you can see here, they are quite diverse in terms of shape or colors. And I just promise you that they look really much nicer if you use a magnifying lens to look through them. So mosses were the first plants colonizing land, so they are very old plants in terms of evolution. And I just want you to make sure that what mosses are. So mosses are plants and they can live together with other plants, as you can see here. And they can also live together with lichens. But mosses here are just highlighted with the red arrow, because mosses, they don't produce flowers, as you might know, they just reproduce through tiny spores. And they are not lichens, because lichens are fungi, let's say mushrooms. So they can live together and they share the same habitat, but they are not the same thing. So mosses are present in all forests in the world, but what did they do exactly? So in northern forests, like the forest we have here in Sweden, mosses played quite important ecological roles, at least 5. And it's especially these mosses forming large carpets on the forest floor who are the most important. So for example, they retain water, so they keep the forest floor humid. They are the unique source of nitrogen in the forest because they are able to establish like close associations or symbiosis with bacteria that fix nitrogen. So mosses are the only input. They provide the only input of nitrogen in the forest. So we can tell that they are natural fertilizers of the forest. These two functions, water retention and nitrogen fixations, are the most important functions for the establishment and development of other plants and trees. And these mosses also serve as a house or habitat for very small animals that then are in the food web. So they are important they have to provide a house. And they are also sometimes food for some specialized animals, like this small lemon. But in general, not many animals eat mosses. So you might know that mosses are very sensitive to environmental change. And this is because opposite to vascular plants, mosses, they don't have roots or tissues to conduct and to transport water and nutrients within the plant. And that's why they are also so small, because they don't have these conducting tissues. So instead, they absorb water and nutrients directly from the environment. So they just absorb water through the tiny leaves. And their growth depends on a very delicate balance of moisture, temperature, and light. So because of this tight relationship with the environment, when the environment is wet, mosses are wet, but when the environment is dry, mosses are dry. So when they are wet, they are active, they can make photosynthesis if the temperature and light conditions are appropriate. But when they are dry, they are completely inactive. They are still alive, but they don't do anything. And this is important to know because when they are wet, they are quite sensitive to temperature or light fluctuations. But when they are dry, they don't care that much about temperature or light. So, and in general, this is not for all mosses, but in general, they like to be wet for a while and then dry and wet and dry. So they don't like so much to be wet for very long periods, and they don't like so much to be dry for very long periods. And in general, in our northern forest, mosses are adapted to cold. So they prefer colder conditions than hot conditions. And I'm saying this because if they are so sensitive to environmental change, how will you manage with climate change? We know that climate change will lead to increased temperatures at a global scale. Climate change will also change patterns in rainfall, and we will have more extreme like climatic events. Like for example, we will have longer droughts, like dry periods, that might be negative for our mosses. So these changes in temperature and maybe water availability might cause that mosses grow less, and eventually they might just disappear from some places. So if this happens, these negative impacts on mosses will have a negative impact on forests. So there could be alteration of forest functions, that, as I mentioned before, like water retention, nitrogen fixation. So these are functions that are important for a whole ecosystem. However, this is just an hypothesis. It's quite logic. It makes sense, but the problem is that we don't really know what's going to happen. And this is mainly because we lack information. There is not data enough to know that this is going to happen. So for this reason, my colleagues and me, we wanted to test like how the same moss can respond to different climates. So we did transplantation experiments using these species that I'm not sure if you know, like Myrhus mossa, Hylocomium numbratum. It has a very wide distribution across Sweden, although its abundance and frequency increases towards north. So we say that it's a northerly species. It's also special moss because it really likes humid and very shady places within forest. So for example, like this is a northern slope in southern Sweden, and here the moss occurs on the forest floor, but also it's covering all these boulders. And as you can tell from this picture, this is a very humid environment, as there are a lot of ferns and other vegetation indicating like high humidity and cold, therefore. So what we did in our transplantation experiment, we took mosses from the south of Sweden and mosses from northern Sweden, and we transplanted them in 30 forest sites in this green square. And these sites were quite special because we selected them in a way that they represented like very different microclimates. And I guess everyone knows what is microclimate. But even if you have been like, let's say, like trekking in a mountain, when you are in the low part, you feel warm, but as long as you go to the top, it's colder. It's just the effect of elevation. But also in a forest, if you, let's say you are in northern slope with a lot of trees, so tree density is high, and then you have a lot of shade, you feel cold. But if you move to just a forest, let's say in a southern slope where trees are more sparse and then the canopy is not so dense, you feel the sun rays on yourself, so you feel warm. So the same happened to our moss. We put southern and northern moss in the same spots where we had like a lot of variation in elevation, slopes, and different types of forests. So our southern and northern mosses experience a lot of microclimatic variation. So for example, they have a lot of variation in soil moisture that we measure with this device. They also experience a lot of variation in temperature, but also in snow melt date that we measure with these temperature loggers. And of course, they were subjected to very different light regimes in very close forest or in very open forest. So this is an example of a transplantation site, and here you can see our transplants, and we can tell this is a northern slope and it's a humid habitat. And this is just like a close-up or zoom of a transplantation unit. So to evaluate the effect of different temperature and climates on our moss, we measure growth and survival of these shoots. And I brought plenty, I will show you. I brought plenty, so if you want to see later, if you want to see it by yourself, you can do it, but there are now too many to do it. But this moss, it has a very funny way of growing. And well, I didn't bring our special moss. I just brought like a very related moss, whose mossa. And these mosses, every year, they produce a segment. And then the next year, they produce another segment. On top of this segment, they produce another segment. So it's very easy to record annual growth. So we just did that. And if you see like in the green part, it means that the moss is growing, but then in the under parts, it starts to decompose. so just get brown. It has a very quite dynamic growth pattern. So if environmental conditions are good for growth, these segments will be very long. But if environmental conditions are bad, these segments will be short, or they might not even come out. So just to make sure that we were following the same suits every year during the experiment, we marked the beginning of new segments with these plastic bits that I guess you might be familiar with. No, this is vegetative reproduction, so it has no spores. This is called vegetative reproduction because it just replicate modules of the same. And sometimes they can reproduce by spores, sometimes. But this species in particular is quite clonal or asexual, so we didn't have data on sexual reproduction on spores. It's just replicating. So we were just monitoring the growth of each segment in all transplants. So this is how it looks like. And I have a recommendation in case you want to do similar experiments, never use the red bits, because for some reason, I guess that maybe birds or other animals, they pick them all. So we lost all of our red marked suits. So what did we find? After 3 years of experiments, what we found is that southern and northern mosses, they do better in the climates that are more similar to their original climates. Here you have like moss performance is a measure of growth and survival together. And here we have temperature from cold to warm. And as you can see here in blue, we have the northern populations. And at colder places, they perform really well. But they do really bad in hot places. And it was the opposite pattern for southern populations in red. So they did much well in hotter spots, but they were very unhappy in cold spots. So it seems that these mosses were adapted to their local climates. So what's the implication of these results in a climate change context? Well, it's not easy to extrapolate, but we could tell that if temperature increased too much, our northern mosses wouldn't be well, because they're very sensitive to hot temperature. And we can't say, even if the southern population they did much better under hot conditions, we can't say that they were happy with that, or that they will be very well if temperatures increase too much. Because also, if you recall, our transplantation area is in northern Sweden, and then we might have more extreme temperatures, for example, in summer in the south. So we can say that climate change will be good. Maybe they tolerate better and under a certain point. And it also depends on how the precipitation will change, that we couldn't control that. So it's still tricky. There is not like a straightforward answer with this. So just to conclude, I just want you just to take to your home that the same species, it's important to know that the same species, even if it's a moss, even if it has like clonal reproduction, they might respond in different ways to climate change. So it's important to take into account this variation within species. So still, we have a lot of uncertainty. We need more experimental studies just to try to predict how these mosses will respond to climate change. And I also want to insist that what I'm presenting here is just a study with one species in one site, and we cannot generalize that all species will behave in the same way. So we really need more people doing more work. And just, I want to highlight that it's really important that we are able to understand how mosses will respond to climate change, because the health of our forests and forest functioning really depends on them. So I just hope that you learned something. I'm quite happy to answer any questions, and thank you so much for your attention.
During which period it was the most mass of the moss?
The most mass?
Mass as a, which period was it in the carbon time? It was most of this bands?
I-
Or during which time?
I don't recall. It's quite, I guess it's even before. This comes before ferns. You might know that better.
Yes, I mean, it would probably be in the Devonian period. I don't have knowledge of the levels, but before mosses had other competitors, they probably would have had a much higher biomass. So plants colonized land in probably the late Devonian. They were really widespread across land. So that's like, yeah, 300 and something million years ago.
Oh, yeah, yes.
Yeah.
That's it.
Yeah.
Okay. Thank you.
Good that you were here. Some more questions?
Are there any more studies now?
Sorry,
Are there any more studies now?
No, we finished this like last year. But I think we should do more.
Would you say that southern mosses better adapted to climate change?
I guess they are adapted to a warmer climate, but also they are adapted to a longer photo period. Maybe they are adapted also to different humidity regimes compared to northern Sweden. But we can't say that they are adapted to climate change. And also, since you might know that when plants reproduce sexually, so they produce like seeds or spores, they just have like, they improve genetic diversity. But in our case, our moss is just reproducing clonally, so we can speculate, this is just pure speculation, that its genetic diversity might not be so high. So then it might struggle to adapt genetically to climate change because of that. So they might acclimatize, so maybe they grow less, but they don't disappear. But I think it will be hard for them to adapt genetically to the change.
Does the species not have any sexual reproduction at all?
It's not at all, but it's very rare. You need like female and male plants, and we don't really know why it's so rare.
Yeah, you mentioned adaptation. How long does it take for that community to adapt to those new conditions? How did you transfer it? By cores or by just by lawns, or how did you do it?
For a community.
Yeah, well, it is a community, even if you're just what you're taking out. I don't know if you use cores and transplant cores.
For the transplantation, we just took mats.
You took the mold.
Yes,
Okay, that's fine.
we took this.
How long does it take? Well, that's a community. There's more to that than just mold.
I would say it's a community if there are different species. Otherwise, it's a population.
Well, yeah, then you've got one species dominant plant,
Hmm.
communities and all that. How long does it take them to adapt to the new temperature regimes?
I don't think it's known.
Yeah, because I mean, you're suddenly moving something that's got maybe a 2-degree change in mean annual temperature.
What it matters,
Suddenly.
yeah, but what it matters the most is not the mean temperature. What it matters is like maximum temperature in summer, for example, in our case, or the snow melt. So that's what is like maximum. So
You don't know how long it takes for those to adapt to the
no, and I guess that they will not. It depends also on the pace of the change. I mean, if it's 2 degrees in 2 years, of course they will not. If it's 2 degrees in, I don't know, 2,000 years, of course they should. Like nitrogen deposition.
Yeah, because you took samples from zones of blood deposition and put them into zones of light deposition.
Yes, that's true. Yeah, no, but it's true that we haven't control for that effect. Of course, there is a effect of the previous environment, but it's also true that we had our experiments for 3 years, and then you usually, if there are this influence of we call it maternal effect. If we have this effect, we should have seen it maybe like the first or the second year. So, but of course, there might be an effect. But no, maybe not. But ideally, but that's why people do not work with mosses so much, because it's very difficult to get a first generation in the lab. When you have plants, you can collect seeds and you can produce your own seeds. And then you know that your seeds have been grown in control conditions. But with mosses, you can't. That's a pitfall of all experiments just using plants that have developed in their sites. Of course, they have a signal. It's not only genetics. They have the signal of their previous environment. Hmm. So there are some other curiosities or? Thank you.